Okay, welcome everyone. I am Istvan Csabai from Eötvös University, Budapest, and I was part of the ELTE complex team together with my PhD student Bálint Patraki. And I will talk about dealing with high-dimensional data to predict preterm birth. So first of all, in the challenge, the preterm birth should have been predicted, and preterm birth means that the birth happens before week 37. And there are two major types of this preterm birth: the spontaneous preterm labor and delivery with intact membranes, shorthanded by SPTD, and the preterm premature rupture of the membranes, PPROM. So actually, we do not know the real causes, what is behind preterm birth, but there should be many complex features. Many may be biomolecular, physiological, external, or stress may cause preterm birth, so there may be psychological, mental agents that may cause it. For the data scientist, this is a problem. So it is infeasible to observe all the real causing phenomena. And also we do not know the model, like we know how planets go around the sun by the Kepler laws. We are way far away to know such a detailed model for preterm birth. So that's why we use the hypothesis that this complex multi-level set of causes have projections into gene expression space. And actually, this multidimensional, in our case, it's about 30,000 dimensional gene expression space, is rich enough, and we can measure the expression values precisely enough to represent the real causes. And also, we assume that from this high-dimensional data set, machine learning methods can extract the true relations. Beyond these hypotheses, there is also a hope that we not just able to predict preterm birth, but also we can find some kind of meaningful interpretation of the selected top genes. Actually, it was part of the challenge description that we needed to develop parsimonious models at most 100 unique genes from the 30,000 in total. There are problems with this also, because not only the birth-related aspects of physiology are projected onto the gene expression space. As you may see on the figure, many other phenomena like age, race, hair color, body mass index, diet, diseases, and you name what, have also some projection into the gene expression values. Also, not just the physiology of humans, but also there is this dark cloud of the observational process that may also bring in various artifacts depending on the type of the chip, the various wet lab parameters, and so on and so on. And in our case, an even bigger problem is that, and this is quite usual for many data science challenges, that the number of samples is only 400 something for the training set, is much, much less than the number of features we are talking about. And we do not know what are the actual numbers of the confounding factors, and that may be also counted in hundreds. And we would like to use this data set to recover real meaningful biological information. So concerning the artifacts, a hard problem for this challenge was that the training set contained two cohorts. One cohort was from a Calgary sample set, and they used HG 2.1 ST gene chip array, and it consisted 320 samples. And the other cohort was from Detroit, and it used another type of gene expression chip, the HTA 2.0 array, and it contained a little bit more than 100 samples. The problem was that the test set for which we should make the predictions had data only with the HTA chip, which meant that part of the training set was biased from the viewpoint of the test set. Also, as you may see on the figure, on the horizontal axis, you can see the gestational age, that there were different sampling types for these different cohorts. And because the rules of the challenge was that we had only 2 submissions allowed, and as we see, the number of samples is relatively small, we didn't have too much space for experimenting with these batch effects. Beyond these biases, we discovered something that was actually important for the final results. So we have discovered that a metaparameter, the gestational age, that leaked some information into the data set that could be used by itself for the prediction. So the organizers of the challenge gave us this information, but actually this was necessary because in the first part of the challenge, we actually shown that there is a correlation between gestational age and expression values, gene expression values. That's why we needed this to use for the second part of the challenge. So it brought in several biases, like the sampling times were different for the full-term and the preterm risk pregnancies. Also, the number of samples per patient differed for the full-term and preterm cases. This is understandable because for those patients for whom preterm birth was suspected, the doctors made more investigations, collected more samples. The third problem was that the last sampling time also was quite different for the different groups, which meant that actually only based on this small metadata, the timing of the samples, we could actually get some information and make some predictions. Actually, this is what we have done, and we used one of our submission possibility from the 2 to make an invalid submission, on purpose invalid or fake submission. And for this submission, we used only this metadata, the number of samplings, and the gestational age at the last sampling. We used quite simple methods like logistic regression and random forest. And surprise, we scored a very high value, above 0.9, and actually we get to the second place on the leaderboard with this very, very raw information without using any gene expression. This meant that there is an information leakage. So our strategy was to make some safe solution, because even though direct usage of this metadata was not allowed by the rules of the contest, but these data may leak into the expression data, and the clever machine learning algorithm may pick up this hidden signal. So we have chosen a fail-safe strategy and sacrificed most of the data to avoid the information leakage. So we used only data from one chip, which already reduced the number of samples to a quarter of it. And since each patient had several samplings, we used only the last samplings. Altogether, we discarded more than 90% of the information, and we used that for the final prediction. And because the training set was very, very small, after all these careful discarding of possibly leaked information, we used a very simple, robust method, random forest, and used this for submission. And this very simple method, which with this relatively small part of the training set, earned us the first place on the leading board. So as some final thoughts, we also, as many other data scientists, use dimension reduction method, as we have done in this challenge also, then the number of features is much larger than the number of samples. And we do this for 2 reasons. One, to avoid overfitting, but I think that modern machine learning methods are fast enough and have robust regularization built in, that this is usually not a concern anymore. The other problem is more philosophical, that we want to get some meaningful causative features out of this complex projection of the data. But since this data comes from a strongly coupled complex systems, this may be a vain hope. Below the pictures show an example from another study where we had about 200 colorectal samples, and the colors mean the different stages of the disease. And when we used the originally more than 50,000 gene expression values and projected with principal components onto the first two components, then we can see that the colors are nicely separated, which means that we could capture the content of the data. But the catch is that if we choose not all the expression values, but random 1,000 or random 100, not the top genes, but randomly, then we can see the same structure. This means that actually from this high-dimensional gene expression data with careful isolating of the artifacts, prediction is easy, but understanding the underlying features is much hard. Thank you very much for your attention.